back to our channel. This is Science Lectures 256. Don't forget to like, subscribe, and hit that button. Also share with your friends this insight of knowledge. So last time we looked at plants and we said we have two types. We have the plant kingdom, and the plant kingdom we said is divided into two. The cryptograms and the phanerogams. And today we are going to look at the cryptograms. We shall later venture into the phanerogams as we are continuing to trace for the development of the plant kingdom. So we say cryptograms. Cryptograms come from two Greek words: crypto, then grammie. And crypto means invisible sex. So these are plants that have invisible sexy organs, or it is very difficult for you to see their sexy organs. In fact, sometimes grammie can mean marriage. So whatever we are going to see here, these ones don't bear seeds. In fact, the other name for cryptograms is lower seedless plants. So all cryptograms do not bear seeds. Let's look at the general characteristics of cryptograms. General characteristics of cryptograms. Number one, they are thalloid. So we said also algae is thalloid, that its plant body is not differentiated into stems, roots, and leaves. So most of the cryptograms are thalloid. Their plant body is not differentiated into that, except for pteridophytes. We are going to see later what are pteridophytes. Then number two, most of the cryptograms are non-tracheophytes. They are non-tracheophytes. So being non-tracheophytes means that they have no vascular bundles. So the vascular bundles, as we saw earlier, are made up of the xylem and phloem. So since they are thalloid, they have nowhere where they could house these vascular bundles. So they have no xylem, and the primary role of the xylem is to transport water, and the phloem transports manufactured food out from the leaf to the rest of the other parts. But we say they are thalloid, so they don't have stems, they don't have leaves. So these xylem vessels will cease function. Still, except there is an exception of pteridophytes. For the pteridophytes are thalloid. They are tracheophytes. And we say, I've just said that being a tracheophyte means that it has vascular bundles, which vascular bundles are both the xylem and the phloem. Then number three, as we said, they are lower seedless plants, so they do not bear seeds. They have no seeds. They do not bear seeds. They have no seeds. Then they exhibit alteration of generations. So alteration of generations is the existence of a plant life in two distinct forms. That is the sporophyte and the gametophyte. The gametophyte. The gametophyte. As you hear, gametophyte, so this is a reproductive phase, and this is the asexual phase or the vegetative phase. And the duration in which a plant spends in these two phases varies from one class to another, as we are going to be seeing. Then they have a motile male gamete. All cryptograms have a motile male gamete. And since, as in animals, we saw that they have motile male gametes. That's why they need water for fertilization. Also, these cryptograms have a motile male gamete. Therefore, they need water. They need water for reproduction. That is why the next point is they are located in damp places. The reason why they are located in damp places is to make sure there is water, which water is needed for reproduction, as they have a motile male gamete. So basically, these are the characteristics of all cryptograms. They are thalloid, non-tracheophytes. They have no seeds, exhibit alteration of generations, a motile male gamete, and they are located in damp places. So cryptograms are divided into two major subphylum. That is, we have our cryptograms here. So they are divided into bryophytes, which are considered to be the most primitive plants. Then they are also divided into pteridophytes. So the bryophytes are further divided into two classes. Number one, there is class hepatica, and class musci. Then the pteridophytes are divided into various classes. There is what we call Cynopsida, Lycopsida, Pteropsida. So we are first going to look at the bryophytes, and later we shall look at the pteridophytes. And the most common example here in the Pteropsida are what most people know as ferns, but there are other types of pteridophytes, things like ferns, whisker ferns, things like club mosses, all fall under pteridophytes. And under class hepatica is where we find an organism called a liverwort. Here we have what most people know as moss, or in plural what they call mosses. So this is the general divisions of both cryptograms. We say they are bryophytes and pteridophytes. Bryophytes is where we find hepatica, where we find the liverworts. Musc is where we find the mosses. The pteridophytes are divided into many: Cynopsida, Lycopsida, Pteropsida, and the likes, as we are going to be seeing. So after having seen cryptograms, now we look at the bryophytes. We say the bryophytes are where we find the moss and the liverworts. Those are the common examples under this subphylum. And we first look at the characteristics of bryophytes. And all the primitive characteristics we've just discussed in cryptograms are majorly based on bryophytes. That means they are thalloid, or their body is a thallus. And we said being thalloid or having a thallus means that their plant body is differentiated into stems, roots, and leaves. Number two, they are non-tracheophytes. All bryophytes don't have vascular bundles, and the vascular bundles we said are both the xylem and phloem. Then they are They are the first embryo phytes. They are the first embryo phytes. And the first embryo phytes. So as we're discussing algae, if you've not watched the videos about algae, please do so. The link is just in the description box below. So as we're discussing about algae, we said algae is a non-embryo phyte because fusion of its gametes, there is no fusion of gametes. So, but these plants are embryo phytes because whenever you have fusion of their gametes, we shall form our embryo. Next, they have rhizoids. They have rhizoids, not roots. Not roots. Why are they called rhizoids, not roots? Remember, the primary role of a root is for both anchorage and nutrient absorption. However, these root-like structures in bryophytes only play a role of anchorage but not absorption of nutrients. Then they exhibit alteration of generation. Alteration of generation. Alteration of generation. And this alteration of generation, as we said earlier, is characterized by having a sporophyte, and a gametophyte. However, the sporophyte of bryophytes is parasitic. Is parasitic on the gametophyte. So the sporophyte depends on the gametophyte. It can be fully parasitic, or it can be hemiparasitic. And let's talk of the sporophyte. The sporophyte of bryophytes is divided into three parts. Number one, the foot, the seta, and the capsule. And we're later going to be seeing the role of each part. But as I've said, the sporophyte is dependent on the gametophyte, and that dependence might be fully or it can be hemi, which means half. Then, these are the general characteristics of bryophytes. Yeah, before I forget, we said the sporophyte, as you hear sporo, it is the spore-producing phase of a plant, and the sporophyte has a group of hygroscopic, spindle-shaped cells called elaters. And the role of these elaters is to aid in spore formation, dispersal. The elaters aid in spore dispersal. That is the role of the elaters. And we are not talking of the reproduction. So these bryophytes have different methods of reproduction. Sometimes they reproduce by gemma formation, and others can reproduce vegetatively. We shall dwell more on that. However, some exhibit sexual reproduction. And the sexual reproduction in bryophytes is ugamas. Sexual reproduction in bryophytes is ugamas. And we say algae has three different forms of sexual reproduction. Can either undergo isogamy, anisogamy, or ugamy. But these bryophytes only undergo ugamy. If you've not yet seen the video talking about the reproduction in algae, please do so, because it is very important for you to understand this. So we say in ugamy, we have both an ovum fusing with an anthrazoid to form an oospore. And this oospore in this case is our embryo. Yeah. So since sexual reproduction is ugamous, that means these plants tend to have specialized reproductive structures. And I want us to talk about the reproductive structures in these cryptograms. However, you want to know that the reproductive structures are produced in the gametophyte phase. the gametophyte phase. The gametophyte phase. What are the gametophyte phase? So the gametophyte, as I was saying, it contains the cells that produce, that later become the specialized reproductive structures. And these organs are surrounded by a layer of sterile cells. They're surrounded by a layer of sterile cells. And these cryptograms have two types of reproductive structures. Number one, they have what they call the archegonium, and they have what they call the antheridium. So this is our antheridium and this is our archegonium. Let's begin with antheridium. The antheridium is a haploid structure. Number two, it is a club-shaped structure. When I say club-shaped, those of you that enjoy games like baseball bat, so the antheridium looks like that baseball, especially the upper part of the baseball. So it is a club-shaped structure. Then it contains, that produces the antherozoids. So we say the antherozoids are the functional sperm cells of these cryptograms. Then the antherozoids are produced from the androcytes. So these androcytes are the sperm mother cells. Or what you could call the stem cells of these antherozoids. Then these antherozoids are biflagellate. They are biflagellated. So when I try to draw the structure, it looks like this. It has two flagella. That is why they say it is biflagellate, and we already say that it is motile, so that is why it is found in areas of damp places. So, and this antheridium has a layer of a jacket of cells which we call the sterile cells. And these sterile cells are what we call the jacket cells. So if we are trying to label this, so this is what we call an osteol. We shall later see the role of the osteol in reproduction. These are our androcytes, which we said are the ones that undergo meiosis to form the antherozoids, which antherozoids are the functional sperm cells of cryptograms. And this is what we call a stalk cell. So then these are our jacket cells. So I hope you can see, I said it is a haploid shaped structure. A haploid structure that is club shaped in nature, has a layer of sterile cells which we call the jacket cells. Then with it has the sperm mother cells which we call the androcytes. We say these androcytes undergo meiosis to form the antherozoids, which are haploid in nature. Then we can look at the archegonium, and this is the male reproductive organ of cryptograms. The male reproductive organ of cryptograms. Now, if we are to look at the archegonium, it is also haploid because it produces a haploid ovum. So its variety is flask shaped. It is flask shaped. I hope you can see the size of the shape of a round bottom flask. It is a flask shaped. Then variety, it contains two parts. It contains the upper part and the lower part. And the upper part is what we call the neck. The neck, and the lower part is what we call the venter. Then this neck contains two types of sterile cells. It contains the neck cells, which are these ones outside here, neck cells. Then it also contains the neck canal cells. cells. The neck canal cells. Which are these ones inside the canal? These are what we call the neck canal cells. The neck canal cells. Then, and the neck is elongated. The elongated part of the archegonium. Then the venter is the enlarged part of the archegonium, where it contains the venter cells. The venter cells. These ones outside here are what we call the venter cells, or when you read some text, they will be calling them the venter canal cells, but they are the same. Then still, this venter will contain the ovum. This one inside here is what we call the ovum. Which ovum is our functional egg cell, which will fuse with the antherozoids to form an oospore, which oospore is our embryo in this case. But I repeat, we said this one is a flask-shaped structure divided into two parts. This is the neck, and this is the venter. The long and slender part is what we call the neck, and the enlarged base is what we call the venter. The neck is made up of the neck cells and the neck canal cells. Then on top of it here has what we call the lead cell. And the venter is the expanded portion of the archegonium. It's made up of the venter cells, or what some texts will call the venter canal cells, and the functional ovum. So that is it for reproduction, and these are the reproductive structures in bryophytes. We shall later see how they operate when we're looking at alteration of generation. After having seen both structures, I want us to compare and contrast by beginning with the similarities between both structures. Number one, they are both haploid. They are both haploid in nature. And being haploid means that you contain half of your chromosome number. Number two, they both produce gamete cells. Then they both mature in the presence of water. And shall later be seeing the importance of water. Then they are both involved in reproduction. And they are both born on modified stalks called gametangiophores. Yeah, these are some of the similarities. Before you forget, they both contain a layer of sterile cells. Both contain a layer of sterile cells. We said that the iridium has jacket cells. The archegonia has the venter cells, the neck cells, and the neck canal cells. So after having seen the similarities, let us look at the differences. The differences between the antheridium and the archegonium. The antheridium and the archegonium. So, as we've seen, this one is club shaped. And this one is flask shaped. This one contains one layer of sterile cells. This one has many layers of sterile cells. This one produces small gametes. This one produces large gametes. gametes. When I say gametes, I mean the ovum. Then see this one produces motile gametes. Motile gametes. This one produces non-motile gametes. Still about the gametes, this one produces many gametes. Many gametes. It produces many anthrozoids. This one produces few gametes. It produces one ovum. You should remember one archegonium, one ovum. One archegonium, one ovum. Then this antheridium forms an ostiole. This one doesn't form an ostiole. So the role of the ostiole is to allow the passage of the anthrozoids from the antheridium so they can swim towards the archegonium. Yeah. Then after fertilization, this antheridium degenerates or it disintegrates. Degenerates or disintegrates. Then this archegonium in some species may persist to form a structure what we call the calyptra. So those are some of the differences between the antheridium and the archegonium. Club-shaped being flask-shaped. Having one layer of sterile cells, having many layers of sterile cells. Small gametes, many gametes. Motile gametes, non-motile gametes. Many gametes, few gametes. Having an ostiole doesn't form an ostiole. Immediately after fertilization, the antheridium degenerates. The archegonium may persist from what we call a calyptra. Lad forgotten, in the antheridium we have no fertilization. No fertilization here. Remember, fertilization takes place in the archegonium. Fertilization takes place in the archegonium. Just like in human beings and all animals, fertilization takes place in the female and the male is just a donor. So that is it for the general characteristics of bryophytes and their reproductive structures. We've seen their similarities and differences. Till next time.